Now we are going to show you determination of total coliform and fecal coliform. To grow coliform, we will need culture medium or broth. Here the darker solution is the broth for the fecal coliform and the lighter solution is the broth or culture medium for the total coliform. We use petri dish to grow the coliform. Before starting the experiment, we first have to disinfect every apparatus that we will be using. We will use ethyl alcohol to disinfect both petri dish as well as the suction pump, also the fork. We will place one filter pad at the bottom part of the petri dish. After placing the filter pad, we will add approximately 2 to 2.5 milliliter of broth in each of these two petri dish. One petri dish will place broth for the fecal coliform and in another petri dish we will place the broth for the total coliform. Then we will place a 0.4 micrometer filter paper and inside the suction cup. After adding the filter pad, we will wash it with the distilled water. So we will add the distilled water and turn on the pump so that the distilled water can be pumped out. The suction cup has also marked for measuring different volume of water. Here we will add 100 milliliter of sample and the measurement will be done inside the suction cup. After taking the 100 milliliter of sample, we will turn on the pump to filter out the sample. Since the pore size of the filter is very small, we need to use this suction to filter the sample. After the filtration, the coliform bacteria will rest on the filter paper. Then we will take the filter paper and place it inside the broth. Similarly, we will filter out another 100 milliliter of sample on a different filter pad, which will be used to determine fecal coliform. After filter out, that filter paper will be placed inside the another petri dish. Then we will allow the filtered papers to rest on the petri dish for 5 minutes so that it can soak the broth. After 5 minutes, we will take out the additional broth from the petri dish and close the lid of the petri dish. Then we will take the petri dish inside the incubator and we will keep there for 24 hours. For total coliform determination, the temperature of the incubator will set at 35 degrees Celsius and for fecal coliform determination, the temperature will be set at 44.5 degrees Celsius. After incubating for 24 hours, we will take out the petri dish from the incubator. After taking out the petri dish, we will observe the bacterial colony. For total coliform, the bacterial colony will be medium red to dark red in color, and we will count the number of colony and we will report the number of colony as colony forming unit per 100 ml. For fecal coliform, the bacterial colony will be blue in color. Similarly, we will count the number of colony for the fecal coliform and report it as colony forming unit per 100 ml.